Welcome to the Basics of Organ-on-Chip Technology 101 webinar, Understanding the Foundations. The first part of this webinar will provide an overview of the historical evolution from 2D and 3D cell cultures to organ-on-chip. We'll mention the challenges associated with 2D and 3D cell cultures, continue with introducing the field of microfluidics and the emergence of organ-on-chip technologies. Further, we'll discuss the advantages and applications of organ-on-chip. The last 15 minutes of this session will be dedicated to any questions you may have, and Revivos microfluidics scientist Philip is here with us today to answer them. Please feel free to type in your questions at any point during the presentation or ask away during the live Q&A. To type your question, please find the chat icon on the bottom panel, or if you can't see it, click More and select Chat. So let's start off with the beginning of in vitro testing. In vitro tests play a pivotal role in scientific research and product development by providing a controlled and reproducible environment outside of the human body. In vitro tests represent an alternative method to animal testing of pharma and cosmetic ingredients and products. For decades, researchers have relied on traditional in vitro 2D cell cultures to understand cellular behavior and drug responses. 2D in vitro tests are essential in investigating biological processes and understanding the underlying biological mechanisms involved in diseases and treatments. As such, they are used in assessing efficacy, toxicity, and safety of new cosmetic ingredients and screening of drugs. But as our understanding of biology deepened, we quickly realized the limitations of standard 2D in vitro models. 2D cell cultures have limited cell-to-cell interactions and lack tissue architecture. Furthermore, the permeability and metabolism of drugs represented in a 2D model can vary significantly from what occurs in our human bodies. To overcome some limitations, scientists have turned towards spheroids and 3D cell culture for their studies. More complex 3D cell cultures involve the use of cells in a gel or scaffold that acts like the natural support structure found in living tissues. Under the right conditions, these cells can grow, arrange themselves, and communicate with each other. The limitation with 3D cell cultures is that the cells lie in a static environment, very different from the continuous movement and dynamic supply of nutrients that occur in the living tissues of the human bodies. Therefore, the question is, how to mimic the complexities of the human body in vitro, bring physiologically accurate features while keeping the system dynamic and alive. The solution to bridging that gap between static cell cultures and in vivo studies is to combine cell cultures with microfluidics. Microfluidics is the study of small volumes of liquid flowing in micro-sized channels, chambers, and cavities. At this state, the fluid enters a laminar, non-turbulent state where the flow is uniform and predictable. By studying the flow and the channels, we can determine the exact properties of the flow and make sure the microfluidic experiments are repeatable and provide reliable and accurate results. Microfluidics allows us to keep a total control over the properties of the fluid, like the temperature distribution, the pressure, and the flow. Hence, we can then recreate the ideal conditions for cells and tissues to grow inside a dynamic system. This is how, in the course of time, starting from 2D and 3D cell cultures, the concept of organ-on-chip was born. By culturing living cells on a highly engineered microfluidic system, we are able to create the right conditions for growing a miniature organ or tissue. Such organ-on-chip is in an environment that mimics our physiological functions and gives us a more realistic view into the inner workings of the human tissues. The features and design of organ-on-chip devices provide several advantages in comparison to static culture. By forming physiology-accurate geometries and precisely controlling medium properties, organs on chip recreate important body conditions like the flow of fluids, the force exerted by these fluids, and how nutrients move around, all of which are essential to keep the cells working well. Furthermore, such technology aims at providing high-throughput screening, which is the ability to perform multiple experiments in parallel. Researchers can then choose specific variables to modify and test, giving them further insight on the model they are studying. Lastly, animal models are limited in their ability to mimic the extremely complex functionalities of human organs and their response to drugs. This is why many promising drugs fail during human trials. Organs on chip, however, are not only reliable, but also cruelty-free and more sustainable alternative method of testing used by the pharmaceutical and cosmetic industries. The applications of organ on chip include understanding the physiology of organs and their related diseases. Researchers can study the intricacies of organ behavior in a controlled setting, observe how diseases develop and progress. Such insights can open up new avenues for the study of complex diseases like cancer, Alzheimer's, or diabetes. Also, organs on chip represent an advanced platform for rapid and scalable drug development. This human-relevant technology can greatly reduce the time and cost associated with drug development by filtering out ineffective or harmful drugs early in the process. Lastly, organs on chip technology can enable the development of personalized cosmetics tailored to individual skin types, genetic makeup, and lifestyle factors. By using patient-derived cells to create, for example, skin and hair models, cosmetic companies can test products on a diverse range of skin and hair types to ensure safety and efficacy across different populations. Organs on chip can be used in a variety of domains such as pharma, cosmetics, chemicals and nutraceuticals research. Finally, let's zoom into the three key features of an organ-on-chip device. The microfluidic channels. The microfluidic channels enable dynamic culture of cells and tissues. The flow of media and reagents mimics the function of the blood and interstitial flow, thus creating a natural human physiological environment in the in vitro model. The tissue model. Tissue models can be real tissue explants or lab-grown tissues based on human cell cultures. Depending on the microfluidic design and application needs, topical and/or systemic treatments are applied to understand their effects. The eluates. Integrating tissue models and microfluidic channels is a powerful approach that allows you to better understand the treatment-tissue interaction by analyzing the soluble factors in the eluates. The microfluidic device that is shown in this slide is Revivo Biosystems Revex chip. We will introduce this microfluidics chip in detail in our following webinar sessions. Stay tuned for more on Revivo's unique organ-on-chip system. In our second webinar, we'll take an in-depth look at the components of our own dynamic organ-on-chip platform and its applications. Thank you for joining us today. Our microfluidic scientist Philip is now ready to answer any questions you might have.